protection from certain threats like maybe heat, maybe radiation, and maybe something. بشكل بسيط جدا السيجنال ترانسدكشن موجوده في كل الليفنج اورجانيزمز starting from المايكرو اورجانيزم اللي هو البكتيريا. فجزء من الاورجانيزمز سر البقاء تبعها انه they are responding to the signals outside, for example by avoiding heat, by avoiding radiation, or sometimes responding to the nutrient concentration. هلا شو اللي بيخلي بعض البكتيريا moving towards certain nutrients in the environment. هي لا بتشوف ولا عندها جهاز عصبي. So it depends on the signal transduction as an action. بالنسبة لنا it could be organismal level or organic or organ level. بمعنى آخر إنه كorganism we respond to these things also by the same mode of action or signal transduction, but in a complex pathway تتطلب تدخل الجهاز العصبي. Inside the body we have organ response and the cell response itself in each organ or in each tissue. So the integrity of single cell within the organism depends on the signal transduction response. هلا إذا ما في response, ما في signal transduction, it could be lethal for these organisms, like human, for example. So basically, responding to the stimuli depends on the availability of the certain transducers in the cells, and eventually we will have certain processes inside the cell. So the stimulus could be hormones, for example, could be light. بهذا الإكزامبل ماخذ group 1 هرمون اللي هم عبارة عن هرمون مرتبط بريسبتر بيعمل complex and it will took the action later on. And group 2 هرمون اللي هم they have many different signals and we have complicated signals in order to reach the final effect. الفرق بينهم انه هذا بيتبع كاسكيد معين لانه مش قادر يروح على النيوكليوس يعمل الاكشن, while here we have certain kind of direct interaction with the genetic material and causing the effect for basically on the gene level by stimulating transcription, for example. So that's why we have transcription factors. So the signal itself is generated once the ligand is there and once the receptor is working. إنه الريسبتر شغال ولا مش شغال بمعنى آخر. Is it normal receptor? In some occasions, the signal could be conducted without the ligand due to the overexpression of certain receptors. So هذا الموضوع إنه when the cell is going to respond to the signal depends على standard condition. And we have ligand and we have receptor. In some diseased or pathological conditions, we have auto or idiopathic response by having active receptors. هذول active receptors either overexpressed معنى آخر موجودين بأكثر تركيز أكثر من اللازم أو mutated. منين فهمنا هذا الحكي؟ من وين عرفنا إنه هذا could be a signaling pathway؟ لأنه mutated معناها إنه يكون defected, which is true. إنه إذا كان الريسبتور فيه defect المفروض إنه ما يشتغل. خصوصا يعني سواء كان في ligand أو فيش ligand. لكن in cancer cases and certain pathological conditions we noticed إنه جزء من هذول mutations will cause constitutive activation بسموها. Constitutive activation معناها الريسبتور always active أو بسبب الميوتيشن أو إنه في عندنا high concentration of that receptor. وأشهر حالة اللي هو HER2 receptor. So the ligand receptor is required in order to have stimulation. And as you know, في عندنا lipophilic group hormones اللي هم they need to go inside to the cell and they have intracellular receptors that might be localized in the cytosol and then move to the nucleus, or we have nuclear receptors by having these stimulators inside or they reach the nucleus actually. Okay, so they are directly affect less than 1% of our genes, but this amount of genes is very significant. على آخر إنه مؤثرة جدا في الأكشن of the cell. مكرر الموضوع إنه في عندنا hormones that are going to inside the cell and they have certain receptors like these receptors, and again the receptor could be inside the nucleus or it could be in the cytosol and it will move to the nucleus. So we are talking about الهرمونز اللي هم lipophilic hormones or hydrophobic hormones. Okay, like the glucocorticoid hormone, for example, or thyroid hormone. So they have different manner of action اللي هو بسبب وجود الريسبتور. Is it cytosolic one or nuclear one? So eventually we have response element. Response element اللي هي RE عبارة عن sequence of DNA that is responding to the thyroid hormone in this case. And here for the glucocorticoid hormone, the letter G is standing for glucocorticoid. So we have different options depending on the receptor type and the ligand type actually. So the gene is going to be activated or sometimes deactivated depending on the signal that's going to be transduced. So we'll have transcription and then finally we'll have the protein. So we are affecting the gene. هلا ما بدي ادخل انا بتفاصيل الجين اكسبريشن itself. هلا الجروب الثاني اللي حكينا عنهم اللي هم البيبتايد and catecholamine hormones. They have cell surface receptors. So the ligands will bind these receptors, and mainly we discussed G protein coupled receptors, and this is the receptor and this is the G protein that is conjugated with the receptor. Once we have the ligand, it will be dissociated and it will cause the release of the alpha subunit that's going to activate certain enzymes اللي حكينا عنهم. جزء منهم الادينين cyclase for example or phospholipase C and so on. هذول كلهم مريت عليهم. Again we have the same list of table that we discussed before اللي هم الهرمونز اللي بيعمل stimulation الادينين cyclase for example or that inhibit in this case الادينين cyclase. That's why they are called I hormones, or S hormone stands for stimulating hormone. برضو مش للحفظ, just to know that we have these groups of hormones. هلا من ناحيه molecular classification الجي بروتين اللي بيكون مسؤول عن stimulation لباثوي معينه it will be called GS, and we have also GI اللي هو standing for G protein or G protein coupled receptor مسؤول عن inhibition for certain pathways, like for example in this case we have inhibition of the adenine cyclase activity due to the presence of this G protein coupled receptor. So it's not always the alpha subunit is activating the adenine cyclase. It depends on the type of the G protein, which is depending on the receptor type. Yeah, they are subtype. When? yeah قصدك الاذر سب يونتس اللي موجودين بالبروتين مالهم. That's because هلا بهذا الاكزامبل احنا مش ذاكرينهم, but they have kind of other regulation types for other enzymes, other metabolic pathways. بس كل اللي اندرسوا كانوا متعلقين بالالفا سب يونت اكتيفيشن لانه هي اللي عندها قدره ترتبط بالجي بروتين. In some references they describe them as just regulatory subunits. في خاصيه بالانزيمز اللي بيعملوا for example kinases. هلا kinases most of them consist of two subunits. unit بسموها C وunit بسموها R. تمام. هلا just dissociation ما صار, واللي بيحفز الديسوسيشن between two subunits هذول لانه هو انزيم بالنهايه مكون من اكثر من وحده, معظمهم مكون من 4 subunits, يعني بتلاقي 2 C's مثلا و 2 R units similar to the G protein. هلا الكاينيز activity موجوده بهذا الانزيم ولكنه inactive because of the attachment to the R subunit, عشان هيك بسموها regulatory subunit اللي بيخلي السي تفك من الار السايكليك AMP for example as a stimulator بيرتبط بهاي الار unit and it will be dissociated. So you will have the active catalytic unit بسموها اللي هي بحالتنا هاي اللي هي تمثل الفا بالنسبه للجي بروتين. So it's very common to have other subunits just to regulate the other part of the protein or the other subunits in the same protein. So that's why ما بنشوف الجاما والبيتا بالاكشن. Okay. بس انا بذكر انه they have certain also regulatory roles in the metabolic pathways. So mainly we are describing here the اللي هو types of alpha subunits that are involved in the stimulation or inhibition, and we have what's called Q and 12 for example. They are just other isoforms of these alpha subunits. Okay, similar to the previous one as a structure in the protein اللي هو G protein coupled receptor. They are alpha subunits, but they are other isoforms احنا بنسميهم, and they are involved in other mechanisms like احنا بنعرف انه في عندنا phospholipase C for example is catalyzed or activated actually by the presence of G protein coupled receptor. Okay, another isoform is called 12 لانه السب يونت هاي يسموها alpha 12 in this example, and it's stimulated by the thrombin. It will release what's called Rho factor, and it's responsible for the cell shape structure. The same example that we discussed before, we have the cell membrane, and we have G protein coupled receptor somewhere here, and this is the adenyl cyclase. الوظيفة تبعته فقط عملية conversion لcyclic AMP وبضل active while the G protein coupled receptor is there as associated form. يعني ما دام الالفا subunit موجودة, we will have cyclic AMP, and cyclic AMP is responsible for the activation of the kinase enzymes. هلا هذا good example على اللي حكيته قبل شوي. We have two types actually of subunits. There are four subunits, but two types لأنه R and C. So once the cyclic AMP is there, it will bind to the R subunit and it will release it. So the C will be free now and it will be active. And once the kinase is active, and in this case it's called A because we have cyclic AMP molecule. So that's why it's called kinase A. It will be responsible for further phosphorylation of many proteins, and these are the sites of phosphorylation. Okay? اللي هم lysine, for example. Serine is not common, but it can be also phosphorylated. But the lysine and sometimes tyrosine are the hotspots of the phosphorylation. بس السيكونس تبعتهم should be in this manner, for example. مش للحفظ برضه. Okay? ففي عندنا tyrosines, في عندنا lysines that are going to be, or sometimes serine are going to be phosphorylated at certain proteins or in certain proteins. هلا مش كل سيرين مش كل لايسين بصير له phosphorylation. It has to be at certain location, basically specific sequence, and then we have the action in the future. هلا هذول I will try to discuss one specific example لأنه بسوى إنه نفهم شو اللي بصير بعدين كexample على kinase activity. So I'll try to figure out a defined pathway. So هلا بالبروكاريوتيك cells زي ما حكينا, just as a comparison story, we have what's called CRP protein that is recognized by cyclic AMP in order to control certain actions. واحدة منهم لها علاقة مثلا إذا بتتذكروا بالجينيتكس تكلمنا عن lactoferrin. Okay, so the lactoferrin is regulated by two pathways or two mechanisms. One of them is the active pathway, and the other one is the اللي هو inhibition pathway إذا بدك. واحد بمعنى آخر بسموها negative, أو واحد ثاني بسموها positive, relying على إنه هل اللاكتوز موجود لوحده ولا اللاكتوز موجود مع الجلوكوز أو الاثنين absent. So we have these two kinds of mechanisms. One of them relies on the cyclic AMP availability, if you remember that. Okay, cyclic GMP is another signaling molecule. It's known as second messenger molecule. And the calcium with the phosphatidylinositol molecules representing another specific example of second messengers. هلا الكالسيوم انا ما بدي اخش في الميتابوليزم تبعه, بس انا بيهمني تعرف انه في عندنا بروتين بالنسبه للموليكولر بيولوجيست it's very common molecule. يعني زي ما كنا نتكلم عن cyclic AMP for example, والcyclic GMP اذا بدك, والادينيل سايكليز, كل هدول صاروا بالنسبه لكم معروفين شو بيعملوا. هلا الموليكولر بيولوجيست they also interested in what's called calmodulin. It's another protein, but this protein depends on the presence of calcium to be activated. Okay, so maybe the name is driven from the calcium involvement. So that's why it's called calmodulin. It's a homologous to the muscle protein troponin C. بيشبه troponin C structure and function. Requires the binding of 4 calcium ions to activate this molecule. هلا هو enzyme basically. And they regulate activity of many structural elements in the cell, including actin myosin complex in the smooth muscle, for example. Microfilament mediation, it means that it will play a role in the cell structure and cell motility as well, like phagocytosis, endocytosis, formation of vesicles, and so on. So this is the molecular structure of the calmodulin. It's one single protein, and it has 2 sites of calcium binding. on the N terminal and 2 sites for on the C terminal. So we have 4 calcium ions that are going to bind this calmodulin structure. So as a cartoon structure, it's represented in this form. So these are the sites of calcium, and this is the calmodulin. And once the calcium is there, it will be activated to have different conformation, and it will bind target proteins in order to cause the action. So the calmodulin itself can be considered as a second messenger as well as the calcium, basically. يعني إذا بدك كل واحد تفصله لحاله, they rely on each other. معنى آخر الكالسيوم لوحده, it's not signaling molecule unless we have calmodulin. So we can consider إذا بدك نقول الكالسيوم هو second messenger or the calmodulin itself. فمش راح تكون big difference بالdefinition. Okay. So the calcium is a mediator for hormone action. So that's why it's called second messenger, لأنه هو متطلب وجوده حتى يعمل activation للكالموديولين. So كيف عرفوا إنه الكالسيوم is responsible or required as a mediator? Because they blunted the calcium from the media, so the media will be free of the calcium, or when the intracellular calcium is depleted. So they artificially causing depletion of calcium. Or sometimes they use mimicking molecules instead of calcium. So they noticed that, or they concluded that calcium is required as a second messenger. Okay. Or influencing cellular calcium flux by stimulators or by other stimulators. كل هذول معناهم انه either انت استغنيت عن الكالسيوم عن الدبليشن او استخدمت agonist او antagonist molecule او عملت calcium fluxing inside the cell by certain other drugs. So all of these mechanisms, لأنه هذول بيختصروا يعني تجارب معقدة جدا how we know إن الكالسيوم is involved in these actions. إما بدك تعمل depletion للكالسيوم أو احنا بنسميه manipulation. تلعب بالconcentration من خلال إنه either flux or influx of the calcium in these cells. So then they discovered that we have different metabolic enzymes that are regulated by the presence of calcium, involving phosphorylation, for example. And all of them are involved in different metabolic pathways like glycogen synthesis, pyruvate synthesis, and so on. So هذول enzymes اللي هم الكينيسيس والسynthesis وإلى آخره are regulated by this action of the calmodulin, either directly or indirectly, depends on the pathway. So this is simple demonstration how the calmodulin is going to work. We have here G protein coupled receptor من نوع Q لأنه هو بيعمل activation للفوسفولايبيز C. هذا مرتبط بالجي بروتين coupled receptor story. Okay. هلا on the other hand we have here the calcium. If you are stimulating the influx of the calcium to the cell, it will be conjugated with the calmodulin and it will cause certain specific action depending on the kinase target of these calmodulin. So the things that we might control by the G protein also can be controlled by calcium, calmodulin axis. Okay. One of the important functions of the calcium is in the mitochondrial metabolic pathways, and this is how we, for example, deliver the calcium to the endoplasmic reticulum. That's controlled also by other hormonal action or signal transduction that will cause the release of the endoplasmic reticulum calcium to the cytosol. So they are interconnected to each other because either we are stimulating the release of the calcium from endoplasmic reticulum or by stimulating the influx of the calcium by the receptor that we discussed before, اللي هو ال Q receptor or G protein coupled receptor from as a type Q. هلا الكاينيز cascade معروفة إنه you are, for example, activating kinase enzymes هون خليني the same thing that we discussed before, activation of the hormones in order to have, or activation of actually signaling molecule in order to have tyrosine in this case, phosphorylation. هذول ال YPs معناها إنه تعمل phosphorylation either للريسبتور أو للsignaling molecule اللي هو insulin receptor stimulating molecule responding to the insulin concentration. or binding actually, and they will cause activation of other downstream احنا بنسميهم proteins like kinases. One of them is the mTOR اللي هو mammalian hormone or sorry mammalian signaling molecule that is known to be similar to the antibiotic resistance gene actually. That's why it's called mTOR, because it's mammalian one. Because we have the TOR one, it's microbial one. So eventually we'll have the same or similar actions by affecting different mechanisms inside the cell. Basically depends on the binding of the receptor and then phosphorylation cascade احنا بنسميها. المشكلة انه في اشياء كثير حاب احكي عنها بس صعب جدا انه الان اطالبكم فيها لانه اذا بدي احكي عنها بدنا نسال عنها. For example, 10 is one of the oncogenes, in another word, that's involved in the development of the cancer if it's mutated in this case. I think you are familiar with this. The PIK is another oncogene because if it's constitutively active, it will be associated with certain cancers, as well as the receptors in this case. So في أشياء كثيرة لها علاقة بالpathology rather than signaling molecule as just signal pathway. هلا هذا الشرح اللي هون كله بيشرح التفاصيل اللي موجودة هون إذا حدا interested بإنه يعرف إيش اللي بصير بالضبط لأنه هاي الباثوي in general one of the most important pathways in the cellular development, apoptosis, cell survival, and cancer development. We have another kind of receptors that are called JAK-STAT receptors, and they are activated in the same manner. We have autophosphorylation of these receptor-coupled proteins. هذول الجاكس بذكرونا بالجي بروتين coupled receptor. بس الفانكشن تبعهم مختلف هون. They are doing autophosphorylation and further downstream phosphorylation. هلا ليش تحكي لنا عنهم هذول كثير او ليش جايبين اياهم كمثال؟ لانه they are very common when we discuss, for example, cell apoptosis or cell survival or cancer. So in the future, you will hear a lot of about the STAT, for example, signaling pathway. Just to be familiar, this is the meaning of the STAT or JAK-STAT pathway. It depends on the phosphorylation or autophosphorylation. The same thing, لما عملنا discussion leptin وال PIK, they are very common in signal transduction. وين؟ associated with many receptors. Thank you. Yeah. هلا حتى الامتور اذا بدك تروح على الريال ايمج او network for this pathway, it's very connected with many other receptors. Okay. هلا جزء منهم inhibitory mechanisms. They are going to inhibit the pathway, like the ptin, for example. جزء منهم is responsible for the activation. بس لان الامور مش بالبساطه اللي بنشوفها هون ذاكرين. يعني جبت هذا السلايد لان ذاكرين معظم البروتينز او signaling molecule as kinases that are involved in the cancer development. عشان هيك انا جبتهم كاكزامبل. اهم واحد منهم اللي حكيته انت اللي هو البي اي 3 كي بروتين اور كاينيز اللي هو الالفا سب يونت تبعته A. Okay, JAK-STAT is another also important signaling molecule that is responding to different hormones or signaling molecules, like, for example, cytokines, in order to have certain responses, including inflammation response. هلا هذا common thing إنه بتحكم بالtranscription, so it's just another simple demonstration of what we discussed in in molecular biology course. هلا هون بتلاحظ إنه هذول المثلثين الصغار هم عبارة عن ligand molecules that are recognizing these receptors in order to cause all of this action inside the cell in order to have gene transcription. يمكن بنتذكر إنه DNA polymerase is not only molecule that is required for the transcription. Okay? أو sorry, RNA polymerase. RNA polymerase هو المسؤول عن transcription process, specifically type 2 in eukaryotic cells. So this stabilized form requires this complex interaction. يمكن سمعتوا قبل هيك بالenhancers والsilencers. So these sequences represent enhancer sequences that are required to stabilize the transcription process. That's why كل ما حكينا عن signal transduction هون زي هدول بالنهاية بالداونستريم بروح نتكلم عن gene transcription. This is because the action of the receptor eventually will cause stabilization of certain transcription unit or transcriptomic unit, like this one as an example. فهذا الرسم عبارة عن توضيح فقط simple demonstration إنه how the hormones are work at the level of the gene. So they are controlling the genes through that controlling of the transcription. خلي الأشياء المرة الجاية.